Hello everyone, I am Dr. Monica. Welcome to my YouTube channel Pathology Learning. Today's topic will be on cell injury and necrosis under the series of cell injury. So let's dive into the topic. In previous videos we saw about cellular adaptations. Whenever there was an injurious stimuli, what happens? Whenever there was an injurious stimuli, the cell will try to adapt to its new environment by adopting certain changes, that is called as cellular adaptations. But when this injury is persistent, then it will result in something called as a reversible cell injury. That is, the cell will get injured, but initially this injury can be reversed whenever this stimuli has been removed. But if this injury is persistent, or if it is or if it is severe, then it will lead to irreversible cell injury, which will then progress and culminate in the cell death. So we'll see one by one about this reversible cell injury, irreversible cell injury and cell death. So what is the most common cause of this cell injury? Most common cause of cell injury is hypoxia. And the most common cause of this hypoxia itself is ischemia. Okay, ischemia is cut off of the blood supply, which will result in hypoxia, and this hypoxia will result in the cell injury. So the organ or the cell which is most susceptible to this hypoxia is, that is, most sensitive cell to this hypoxia is a neuron. It can withstand this ischemia only for about 2 to 3 minutes, after which the cell will die. Next, the cardiac muscle. The cardiac muscle, it can withstand for a time being of 20 to 30 minutes only, after which there will be infarction of the cardiac muscle. Next, the most resistant cell to this hypoxia is a fibroblast. Fibroblast is the most resistant. So all of this has been asked as MCQ questions before. So remember, the most common cause of cell injury was hypoxia, while the most common cause of hypoxia was ischemia, and we saw about which are the cells which are most sensitive and most resistant to this kind of cell injury. So coming on next to reversible cell injury. So in reversible cell injury, the first organ to be affected is a mitochondria. While the first change, remember it is the first change which is happening in the cell injury, will be a mitochondrial dysfunction. We all know that mitochondria is the powerhouse of the cell, right? So the powerhouse of the cell, it will actually produce ATP by the process of oxidative phosphorylation. So in the presence of oxygen, oxidative phosphorylation will happen, and that will result in the production of ATP. This ATP is needed for certain pathways to happen in the cell, which is needed for the survival of the cell. So whenever there is mitochondrial dysfunction, this ATP will not be produced, right? Or it will decrease in the production. There will be decrease in the production of ATP, and that is reduced to 5 to 10% of the normalcy. Okay, this again has been asked as a question. When ATP levels decrease to 5 to 10% of the normal levels, then it will result in the cell injury. So let's see what happens when ATP decreases. So firstly, what happens when ATP decreases? It can either affect the endoplasmic reticulum, the cell membrane, or the nucleus. So what is endoplasmic reticulum? It is the site of protein synthesis. This is the rough endoplasmic reticulum on which we see these ribosomes which are attached. When these ribosomes start detaching because of the decrease in the ATP, so that will result in the decrease in the protein synthesis. That is one of the changes which happens because of decrease in the ATP. Nextly, in the membrane, we'll see what happens. There is a pump called as potassium ATPase pump. So this is dependent on ATP. So when this pump fails because of decrease in the ATP, what will happen? There will be increased sodium influx into the cell and increased potassium efflux. Normally, the potassium is supposed to be inside the cell, while sodium is supposed to be present outside the cell. But when ATP is not there, what will happen? The sodium increased influx will happen into the cell, and the potassium will start going outside of the cell. This increased sodium influx, along with the sodium, there will also be, it will be dragging along water. Okay, so water will also come along with the sodium into the cell, and this will result in something called as cellular swelling. So whenever water enters, the cell will obviously swell, right? So this cellular swelling is the first morphological or the microscopic change PC in a cell injury. Remember, the first change, if they ask in a cell injury, it is mitochondrial dysfunction, while the first morphological or a microscopic change which occurs in a cell injury is the cellular swelling. And the cellular swelling is also called as vacuolar degeneration or a hydropic change. So what will happen when the cell swells? The membrane, it will start producing certain blebs. Okay, so in the membrane we can see the presence of certain blebs, and the microvilli which was present on the cell membrane, it will start losing it, it will start detached, so it will cause loss of microvilli also. Along with the cellular swelling, the organelles will also start swelling, like the mitochondria, endoplasmic reticulum. These organs will also undergo swelling. So there is next change which we see, the most important change is the presence of the myelin figures. So what are these myelin figures? These are nothing but whorled appearance of phospholipids. These phospholipids are actually derived from the cell membrane. Because of cell injury, these phospholipids tend to form these concentric lamellations which are derived from the cell membrane, right? So these are called as myelin figures. These myelin figures can be present both in reversible cell injury and irreversible cell injury. Okay, so it is not specific for reversible cell injury. It can be seen in both in reversible and irreversible cell injury. But remember, it is in the irreversible cell injury that the number of these myelin figures are increased. Okay. Next, coming to the nucleus. Whenever there was hypoxia, right? Hypoxia, whenever there is, oxygen is not available, the cell will undergo anaerobic glycolysis, the end product of which is lactate and pyruvate. So this lactic acid will decrease the pH because it is an acidic product, right? So it will decrease the pH. So decrease in pH will in turn result in the nuclear chromatin condensation or clumping. So the nucleus will clump as end result of this anaerobic glycolysis. So because of ATP, we saw three things which were happening. First one was decrease in the protein synthesis. Next is the cellular swelling and the formation of these myelin figures in the cell membrane. And the third thing in the nucleus which is happening is the nuclear chromatin condensation or the clumping. So moving on, what other changes can be present in a reversible cellular injury? In the mitochondria, we can see the presence of small amorphous densities. Okay, there will be presence of certain amorphous densities which are made up of actually calcium, and these are very small and number will also be very less, and that is seen in the reversible cell injury. Next, coming on to the liver, there is a special finding which we see in the liver, that is called as the fatty change or the steatosis. This happens because of the metabolic derangements happening in the cell. So when the metabolism is getting deranged, what will happen? The triglycerides which are present will start accumulating. Triglyceride accumulation will be there, and that will result in something called as the fatty change or steatosis. So look at this image. Can you see? The normal liver is supposed to be pinkish and granular. Okay. So in this, what we see, it is all having some vacuolations and clear spaces, right? So can you appreciate this one? It is having multiple vacuoles in its cell. If suppose this was the nucleus, if we think, then there are these multiple vacuoles which are seen inside the cell, and that is called as microvesicular steatosis. However, if you see this cell which is big, like it is looking more like an adipocyte, there is only a single vacuole and the nucleus has been pushed to the periphery, and this is called as macrovesicular steatosis. Okay. So in reversible cellulary, there are two characteristic changes, both the cellular swelling or the hydropic change and fatty change or the steatosis. These characteristically appear in a reversible cell injury. So moving on, let's see some other images also. So any of this can be asked as an image based question. So what is this first image? If you see, this is the image of a kidney actually, showing these renal tubules. These are renal tubules. Compared to this renal tubule, if you see, it is all quite enlarged, and the cells also, if you see, it is quite vacuolated. They are swollen up and they are vacuolated, right? Swollen up and the vacuolated. So what is this? This is nothing but the hydropic change or the cloudy swelling, of the kidney. So this was one of the reversible changes we saw. Coming to the next image, this, what do we see? Can you appreciate that this structure is all it is all lamellated like this, right? Like an onion skin it is appearing. So this is nothing but a myelin figure which is composed of the phospholipids derived from the membrane. And these are characteristically present both in irreversible and reversible cell injury, but the number of which is more in the irreversible injury. So moving on to irreversible cell injury. The characteristic features of irreversible cell injury is two M's. Remember both the M's, membrane and mitochondria. These both will be damaged in a irreversible cell injury. So membrane and mitochondria damage, that will result in a irreversible cell injury. We'll see what happens in a membrane damage. Whenever the membrane is damaged, right, the cell will open up. The cell will result in death, obviously. When the membrane is damaged, it cannot survive. So it will result in a form of cell death called as necrosis. So in necrosis, remember, the cell membrane is always damaged. And whenever the cell membrane is damaged, what will happen? The contents of the cell pour into the environment, and that will attract the inflammatory cells like neutrophils and macrophages. So it is always associated with inflammation. So remember, necrosis is a type of cell death in which there is membrane damage along with the presence of inflammation. So let's see what happens when the membrane is damaged. When the membrane is damaged, the calcium which was present outside the cell, it will enter into the cell. So that is called as calcium influx. So when calcium enters, it is activating the enzymes which are present. So what are the enzymes which are getting activated? Firstly, the phospholipase, which can damage the membrane. So phospholipid is present in the membrane, right? So it will damage the cell membrane. Then protease, it will damage the cytoskeletal proteins which were present. And next, the nuclease can affect the nucleus. So the nucleus will undergo characteristic changes, which is pycnosis, karyorexis and karyolysis. Remember, it is in this order only you have to remember: pycnosis, karyorexis and karyolysis. So what is pycnosis? Pycnosis is nothing but the chromatin is getting condensed. The nuclear chromatin, it is getting condensed and darkened appearance. Okay, hyperchromatic condensed nucleus is the pycnosis. While karyorexis means something is breaking or fragmenting. So this karyorexis is when the nucleus gets fragmented. While karyolysis is the nucleus itself is lost. It is being broken down. So when the nucleus is broken down, what will happen? The nucleus which was basophilic in color, now the nucleus is not there. So either the nuclear basophilia will be decreased, or the nucleus will not be present at all. So nucleus is lost, or there will be decrease in the basophilia of the nucleus. Right? Remember, in the nucleus, the characteristic changes involved is pycnosis, karyorexis, then karyolysis. So we saw the membrane is damaged now, the cytoskeleton is damaged now, and the nucleus is also damaged now. With all the three loss, there is no point of the cell living. So the cell will undergo the form of cell death, which is necrosis. So this calcium influx can also lead to the formation of large flocculent amorphous densities in this mitochondria. We saw small amorphous densities which were present in the reversible cell injury, right? But here in irreversible cell injury, the same calcium can form very large amounts of these flocculent amorphous densities, and that is characteristic of irreversible cell injury again, okay? And next, moving on to the severe mitochondrial damage. So when mitochondria is damaged, it will result in both the kind of cell death, that is apoptosis and necrosis. So let's see how. Apoptosis, we'll read about apoptosis later in detail in the upcoming videos. There is a pathway involved in apoptosis called as the intrinsic or the mitochondrial pathway. Intrinsic or the mitochondrial pathway. So when this mitochondria gets damaged, it will trigger this intrinsic pathway and the cell will undergo apoptosis. So in for necrosis, just now we saw that when mitochondria is dysfunctioning, the ATP production will decrease. So ATP production decrease, we saw it will lead to cellular swelling, myelin figures, nuclear chromatin clumping, and endoplasmic reticulum protein synthesis getting decreased, right? We saw the effects of decreased ATP. Along with decreased ATP, we also saw the increase in the calcium influx, which was resulting in this form of cell death called as the necrosis. So, mitochondria, whenever it's damaged, it can result in both the kind of cell death, that is apoptosis and necrosis. So let's see some of the images. This is a normal cell, okay? This is the normal cell which we are seeing, and this is the nucleus of the cell. See quite big how it is. Now, in the second image, if you see, the cell is all becoming, this nucleus is all becoming totally hypochromatic. Compared with this first image, you can see that the second image, the nucleus is all clumped and chromatic. It is looking dark in color. So, this is called as pycnosis. Pycnosis was nothing but nuclear chromatin condensation, right? Nuclear chromatin condensation. So, the next image, see, if you see here, the nucleus is all broken up into these small things. So, it is all getting fragmented. So, nuclear fragmentation is called as karyopexis. So, karyopexis is nuclear fragmentation. The third image here is, can you see this cell is just a ghost. There is no nucleus present in this cell. So, what is this? The decrease in the basophilia of the nucleus. The nucleus itself has lost its basophilia, and here there is no nucleus at all. So, this is called as nucleus has broken down. That is called as karyolysis. Any of these images can be given, and it can be asked, you can be asked to arrange in a sequence. Sequence based questions are coming, right? So, these images can be given and you can be asked to arrange in sequence. So, first the change happened was pycnosis, then was karyopexis, then is the karyolysis. So, what is this third last image we see here? This is an electron microscopic image, and these structures are all mitochondria, okay? These structures are all mitochondria, and the mitochondria is quite swollen up over here. The normal mitochondria will not be this big in size. It is all quite swollen up, and inside this mitochondria, if you see, can you see the presence of some electron dense these materials? Here also there is one, here again, here. So, these electron dense materials which are present are the large flocculent amorphous densities. Large flocculent amorphous densities which were composed of calcium, and this is characteristically seen in the irreversible cell injury. While the small amorphous densities were characteristic of reversible, the large amorphous flocculent densities are characteristic of irreversible cell injury. So, we saw how cell death of both necrosis and apoptosis type can happen because of the mitochondrial damage, and when the membrane was damaged, it will result only in the type of cell death which is necrosis. So, this we had done. So, let's see what is the difference between this necrosis and apoptosis. Both are forms of cell death, that far we have understood now. While necrosis is always pathological, it can never happen in a physiological process. It is never happening as a physiological process. It is always pathological. While apoptosis can either be a physiological or a pathological process. While in cell necrosis, we saw that the membrane must be damaged, right? The membrane must be damaged, and when the membrane damaged, like I told, to clear up the debris, it will invite the phagocytic cells like neutrophils and macrophages. So, associated inflammation is always present in a necrosis. And here the first change we saw was a cellular swelling. So, reversible cell injury we saw, right? So, cell will get swollen up and the nuclear chromatin will undergo condensation, then it will undergo fragmentation, then breakage, that is pycnosis, karyopexis and karyolysis. So, these cellular swelling and this characteristic nuclear findings are seen in a necrosis, while it is the complete opposite in an apoptosis. Here, the cell membrane will never get broken up. The cell will actually shrink in size rather than swelling. The cell will actually shrink in size, and the nucleus will undergo something called as a nuclear chromatin condensation. Like a pycnosis, the same thing will happen in apoptosis. But here, the nucleus will not, it will undergo something called as an internucleosomal cleavage. So, the nuclear chromatin condensation and internucleosomal cleavage is the characteristic finding of an apoptosis. And for apoptosis, it is a programmed cell death. It occurs in a regulated cell manner, just like that randomly like a necrosis, it doesn't happen. It is a quite regulated, tightly regulated mechanism controlled by certain genes. And that is why it's termed as programmed cell death. And this is dependent on an enzyme called as caspase. So, the activation of this caspase is the hallmark of apoptosis. While necrosis, we don't see any of this caspase activation. So we saw about the basics of necrosis, apoptosis, reversible cell injury, irreversible cell injury. So in the next video, we'll be reading about the patterns of necrosis, and then later on we'll discuss about the apoptosis pathway in detail. So that's it for today's video. I hope to see you soon in the next video, which is on the patterns of necrosis. Thanks for watching, and if you like my content, consider subscribing, and you can also share it with your friends who might also benefit from this video. Thanks for watching once again.